Good day to all. In today's video, let's discuss about monoclonal antibodies. So generally, antibodies are produced by a foreign antigen enters the body due to the immune response. B lymphocyte develop into plasma cells and it produce antibodies of different types. So types of antibodies here in clonal, it is monoclonal antibodies and polyclonal antibody. So monoclonal antibodies that are identical because they are produced by one type of immune cell, all clones of a single parent cell. So whereas polyclonal antibodies are antibodies that are derived from different cell lines. So monoclonal antibody production. So monoclonal antibody production undergoes a procedure where it includes immunization, cell fusion, selection of hybridoma, screening, cloning and propagation. So when we see about immunization, so immunizing an animal, specifically here we use mouse. So immunizing a mouse by injecting with an appropriate antigen along with an adjuvant. So injecting antigen at multiple sites or repeated several times for increasing stimulation of antibodies. So 3 days prior to sacrifice the animal, a final dose is given intravenously. So spleen is aseptically removed and disrupted by mechanical or enzymatic method to release the cells. So using density gradient centrifuge, lymphocytes are separated from rest of the cell. Cell fusion. So in this process, lymphocytes are mixed with the myeloma cell and it exposed to polyethylene glycol for a short period of time, and the mixture is then washed and kept in a fresh medium. So the mixture contains hybridomas, free myeloma cells and also free lymphocytes. So selection of hybridoma. So the fused mixture is now cultured in hybridoma technology, which is a hat medium for 7 to 10 days. So due to the lack of HGPRT enzyme in myeloma cells, the salvage pathway is not operative, so it will not undergo salvage pathway, and hat medium blocks the de novo synthesis and for nucleotides. So hence myeloma cells cannot survive here. So as the lymphocytes are short-lived, they also slowly disappears. So thus hybridoma are selected using hat medium. So screening is done. So screening is done for antibody specificity to see the specificity of the antibody which is cloned. So the hybridoma culture must be tested for desired antibody specificity. So immunological techniques like ELISA and RIA are used for screening. So in these tests, the antigens are coated to plastic plates. The antibodies specific to the antigens bind to the plates. So the remaining are washed off. So thus the hybridomas producing desired antibodies are identified and screened here. The next process is cloning and propagation. So the single hybrid cell producing the desired antibody is isolated and cloned. Usually two techniques are commonly used for this. One is limiting dilution method and the next one is soft agar method. So in the first method, suspensions of hybridoma cells is serially diluted so that each aliquot of each dilution has one hybrid cell. So in the soft agar method, the hybridoma cells are grown in soft agar. So these form colonies and the colonies are monoclonal in nature. Monoclonal antibodies are identical immunoglobulins generated from a single B cell. So these antibodies recognize a unique epitope or a binding site in an antigen. So types of monoclonal antibodies are murine monoclonal antibodies, chimeric monoclonal antibody, humanized and human monoclonal antibodies. So these are the types of monoclonal antibodies. So monoclonal antibodies are classified into different types based on their origin and structure, particularly the proportion of human versus non-human component, which is usually mouse. So how much it derives and how much from their origins, and based on all this there is different types of monoclonal antibodies. The application of monoclonal antibodies can be broadly categorized as diagnostic applications. So in diagnostic applications, specifically in biochemical analysis, Antibodies are used in several diagnostic tests to detect the toxins, hormones, or hormone related disease. And monoclonal antibodies to HCG are used in pregnancy test kits. And when we talk about therapeutic application, monoclonal antibodies can directly use in the treatment of cancer and in the immunosuppression of organ transplantation, and also monoclonal antibodies can be used in autoimmune diseases and all, and protein purification. Monoclonal antibodies can also be used to purify a substance with techniques called affinity chromatographies and all. So these are all some of the applications of monoclonal antibodies. Thank you.